I was right. I didn't understand exactly why, but I was right all along. Turns out, in my previous video, I barely scratched the surface of how ridiculous the Cosmos trial is. The Cosmos trial was this huge clinical randomized trial sponsored by Mars and Pfizer that lasted approximately 3.5 years with more than 20,000 participants enrolled. Let's count all the giant red flags that the cocoa extract arm of this trial has. In this episode, p-hacking, how to power your clinical trial and what it even means, confidence intervals, p-value and fragility index, relative risk reduction, absolute risk reduction, and numbers needed to treat. Buckle up, it's gonna be a fun ride. The sweet irony is that the Cosmos trial actually proved that if you are a healthy individual, you won't benefit from cocoa extract supplementation. If you are an unhealthy individual with heart problems, there aren't any clinical trials with hard endpoints that I'm aware of to support any claims regarding cocoa flavanols. Mars wants you to pay them $45 on a monthly basis. Let's crunch the numbers. 45 multiplied by 12 months in a year, that's $540 annually. And let's multiply it by 3.6 years, the time that the Cosmos trial lasted. You will pay Mars almost $2,000 in less than 4 years. What are you getting in return? First and foremost, the trial didn't reach statistical significance for the primary endpoint. Cocoa extract supplementation did not significantly reduce total cardiovascular events among older adults. Period. But since it's Mars, fucking giant corporations that exploits 6-year-olds to harvest cocoa beans, they resorted to cheating and added another line to their conclusions, but reduced CVD death by 27%. Let's talk about p-hacking. There are 11 secondary other outcomes listed here. If you run 11 separate statistical tests at the standard alpha level of 0.05, the probability of finding at least one false positive, type 1 error, purely by random chance, increases dramatically, family-wise error rate. If the researchers adjusted for multiple comparisons using a method like Bonferroni or Hochberg, the threshold for statistical significance, alpha, would drop significantly lower than 0.05. Testing multiple endpoints without a rigorous adjustment plan is often referred to as data dredging or p-hacking, as it increases the likelihood that a statistically significant result is merely a product of random chance, rather than a true biological effect. In the SELECT trial for Wegovy, a phase 3 trial, they also had multiple secondary endpoints. Because these supportive endpoints were not adjusted for multiplicity, confidence intervals should not be used in place of a hypothesis test. Your quote highlights a massive trap in reading clinical trial data: multiplicity, the multiple testing problem. If a study measures dozens of supportive or secondary endpoints, e.g. tracking strokes, heart attacks, cognitive scores, and sleep quality all in the same trial, the mathematical rules of a standard hypothesis test start to break down. If you run 20 hypothesis tests at a 5% significance level, by pure random chance, one of them is highly likely to look statistically significant. To fix this, researchers must mathematically adjust their hypothesis tests using multiplicity adjustments to make the barrier to entry much stricter. If they don't adjust for it, any individual positive result is highly likely to be a fluke. By the way, in my previous video, I think I messed up the definition of a confidence interval. A 95% confidence interval shows the range of values within which the true effect likely falls. Even if a 95% confidence interval for a hazard ratio doesn't cross 1, it looks like a win. However, because these specific supportive endpoints weren't part of the strict adjusted hypothesis testing plan, looking at the confidence intervals gives a false sense of certainty. The quote is warning you: do not look at the tidy boundaries of that confidence interval and assume it proves a real effect. Because they didn't adjust for the sheer number of questions they asked, you cannot treat it as a rigorous past hypothesis test. All the highlighted endpoints are below 1 and technically reached statistical significance. But they say again and again throughout their paper, the widths of the confidence intervals have not been adjusted for multiplicity and therefore should not be used to infer definitive treatment effects for supportive secondary endpoints. For my previous video, I had to record 10 sets of 10 coin tosses, and I had a pretty funny one. It was quite unremarkable until the 9th set, when I got 10 heads in a row. The odds of that happening is 1 in 1,000, or less than 1/10%. As far as I understood, it's not common practice to actually adjust p-values and confidence intervals. They are just not reported as definitive. The SELECT trial conclusions sound very modest in comparison to the COSMOS trial. In patients with pre-existing cardiovascular disease and overweight or obesity, but without diabetes, weekly subcutaneous semaglutide at a dose of 2.4 milligrams was superior to placebo in reducing the incidence of death from cardiovascular causes, non-fatal myocardial infarction, or non-fatal stroke at a mean follow-up of 39.8 months. The first giant red flag, the COSMOS trial didn't adjust for multiplicity. They even acknowledge it themselves in the paper. We did not adjust p-values or confidence intervals for multiple testing. Consequently, results for secondary outcomes, other outcomes, subgroup analysis, and other analysis should be interpreted cautiously and considered hypothesis generating. At the nominal 0.05 level, we would expect less than one interaction and one secondary outcome to be significant by chance alone. And yet, they made a definitive claim in their conclusions. That's cheating. I also want to mention why the SELECT trial stopped reporting p-values after death from cardiovascular causes. Confirmatory endpoints are part of a pre-planned, mathematically rigorous testing sequence. They are explicitly included in the trial's formal statistical plan to see if the drug can claim additional official benefits beyond the primary endpoint. They are usually evaluated using a hierarchical step-down testing procedure. You can only test the first confirmatory endpoint if the primary endpoint passes. You can only test the second confirmatory endpoint if the first one passes, and so on. That's why the COSMOS trial doesn't report any p-values, because they failed at the very first hurdle, the primary endpoint. In my previous video, I said that the entire COSMOS trial now seemed underpowered. It was an assumption, but I didn't pull it out of thin air. There aren't that many variables that you can feed in Schoenfeld's formula. Type 1 error is your p-value of 0.05. Type 2 error is typically 0.2. Intervention and control groups are divided equally. These are fairly standard. The last ingredient for this type of trials was hazard ratio. Again, I used chatbot's suggestion that in cardiovascular outcomes trials, the researchers usually set this to 0.85. Meaning that they expect that their intervention will help reduce number of CVD events by 15%. That's how I came up with approximately 1100 events needed for the trial. The only problem is that I didn't understand what any of this means. In the very beginning, when I only started researching for this topic, I asked ChatGPT to help me calculate the power for my trial. It asked me two questions that completely took me aback. How much bias I would allow, and what's the effect size I was expecting. Back then, I just dropped the subject altogether. But over the past week, I had so many conversations with ChatGPT and Gemini. At times, it felt like I'm gonna have an aneurysm or something. I couldn't make heads or tails of it. Until I switched to Claude, and it all suddenly clicked. Not because Claude is better at explaining, but because I don't typically use Claude, and I needed a clean slate. Again, I asked it to help me calculate the power for my hypothetical trial, and it went ahead without me giving it any input, and it was a stroke of luck. This can't be right. I just used Schoenfeld's formula to calculate the power for my hypothetical trial, and the numbers were significantly higher. Something's not right. Hazard ratio went from 0.8 to 0.85, and it doubled the number of events needed for your trial. Suddenly it all became so much clearer. If your intervention can provide modest 5% reduction, I'm gonna tell you right away, it's not financially viable to run a clinical trial to prove it, because you will need, checks her notes, 12,000 events! You see, I argue that the power of the trial means its precision, and I'm pretty sure I'm right. If your intervention has such a weak impact, aka effect size, which in this case is hazard ratio or HR, you will need a highly calibrated tool to measure it. It's like you have a ruler and you want to measure distances in your garden. You don't need millimeters, centimeters, or inches to do that. As a matter of fact, you might not even need a ruler to do that and estimate everything by naked eye and be quite precise. But if you want to know the size of a microbe in a droplet of water, you will need something more precise than even a millimeter. So how come power and precision aren't the same thing is beyond me. Anyway, we actually don't have to guess whether the Cosmos trial was underpowered or not, because they say so themselves in the protocol of the study. With approximately 22,000 randomized participants, there will be at least 80% power to detect an 11 relative hazard reduction in total CVD, and more than 95% power to detect such a reduction in the important composite of total CVD plus all-cause mortality with the expected number of events. You see, turns out my assumption that the Cosmos trial was underpowered was more of an educated guess, really. And initially they had a completely different primary endpoint. The total number of cases of cardiovascular disease, CVD events. Cardiovascular disease events include myocardial infarction, MI, stroke, coronary revascularization procedures, and cardiovascular death, but they had to expand it before unblinding. The expected rates for our original primary composite cardiovascular disease endpoint were based on the projected age and sex distribution of the trial cohort and CVD event rates from our previously conducted trials. However, we determined that the observed rates of CVD endpoints among Cosmos participants were lower than expected due to a younger population of women, increasing use of statins and other pharmacotherapies as seen in other recently published clinical trials, and the impact of COVID-19 on fewer reports of CVD diagnosis, hospitalizations, and procedures. As a result, the Cosmos Data and Safety Monitoring Board approved the proposal to add 3 new outcomes to our primary composite CVD endpoint: unstable angina or acute coronary syndrome requiring hospitalization, carotid artery surgery, and peripheral artery surgery or angioplasty. I'm not saying that they have this low count of events because they actively cherry-picked those outcomes that suited their agenda, and that the real numbers are completely different and far from being so polished, and that all their claims are built on lies. I'm not saying that. But I wouldn't be surprised if that was the case. The SELECT trial also ran through COVID pandemic. Somehow they didn't have such a problem. They even have a separate chart where they included COVID-related events. The Cosmos trial was underpowered even for the primary endpoint. That's huge red flag number 2. By the way, 3-point MACE wasn't even included as an endpoint, and Claude mentioned something else about their protocol. Version 21 is a lot of amendments. For context, most well-run trials end with 5 to 8 protocol versions. Reaching version 21 suggests that the protocol was substantially modified many times during the trial, which raises questions about how much the design drifted from the original pre-specified plan. What protects me from poorly conducted, underpowered studies like the Cosmos trial? Ideally, confidence intervals and p-values. But because Mars Incorporated resorted to cheating and didn't adjust p-values and confidence intervals, we cannot really rely on those. Instead, we're going to use fragility index to counteract their bullshittery. But first, I want to show you how important the number of events really is in this type of trial. Imagine you have a drug that reduces hazard ratio for CVD events by half. You don't need that many events if the effect size is this large. And I actually talked about it in my previous video. You will need only 66 events to prove it. You then have to decide, of course, which cohort of participants you have to put together to see this effect in a reasonable time period. Basically, which target demographic will benefit most from your intervention. That's where epidemiology comes in. Whether your subjects need to be smokers or not, overweight or have normal BMI, whether they have a history of diabetes, CVD, etc. But for our purposes here, it doesn't really matter that much, so I'm leaving everything here as is. And it calculates that we need 214 subjects. As far as I understand, you calculate confidence intervals for hazard ratio a bit differently from how you would calculate it for relative risk reduction, because hazard ratio also takes time into account, i.e. how long it actually took to have a CVD event. But since it's all hypothetical, we're gonna calculate confidence interval for relative risk. Again, this is my personal opinion, but from what I see in this type of studies, low count of event inherently implies low precision, and therefore you get humongous confidence intervals. Math doesn't know how effective your drug is. You ran one test, you got one snippet of what your drug might actually do. Then math makes calculations and assumptions. your drug could average out at potentially reducing CVD events by 63% or possibly just by 13%. Want higher precision? Increase the number of events by enrolling more participants or by running the experiment for a longer period of time. If we look at cocoa supplementation, we see that it might reduce CVD death by whopping 46%. Then Kuna Indians on the islands of the Panama coast would be immortal. Or it might reduce CVD death by measly 2%. And there is no way of telling which one it is. So how is this different from a coin flip? What a larger sample size actually does is protect you from a false effect size. In a small study, the calculated effect size is incredibly erratic. It bounces around wildly because a few deaths here or there distort the percentages. You can easily pull a massive effect size out of thin air just by being lucky. In a large study, the calculated effect size becomes stubborn. It stabilizes. Whatever number it lands on is highly likely to be the actual truth. I understand what p-value does and how it's supposed to protect me. It tells you what the odds are to get this exact distribution of events between intervention and control by sheer luck. P-value for CVD deaths in the COSMOS trial is 0.038. It means that there's 3.8% chance to get this result by luck. Look at the SELECT trial again. They didn't reach statistical significance on CVD death outcomes. Think about it for a second. How come stupid cocoa flavanols can have greater impact on healthy people than a weight loss drug on obese people with pre-existing heart condition? It doesn't make any sense. It has zero biological plausibility. So there is this peculiar metric that can actually demonstrate how brittle p-values are in the COSMOS trial. It's called fragility index. It shows how many events you have to flip to the other side to render the results statistically insignificant. It's 1 for CVD death. It takes one single death and only 3 MACE events flipped to the other side to make this entire thing statistically insignificant. Both of these significant findings rest on differences so small that they could plausibly be explained by chance, minor data handling decisions, or losses to follow up, rather than a true biological effect of cocoa extract. If you look at the SELECT trial, the fragility index is 63. You will need to flip 63 outcomes to the other side to render it statistically insignificant. The difference is so dramatic, you can't not see it. I'm not sure how often fragility index is used in statistics, but for me personally, it's another huge red flag that points in the direction of a poorly conducted, possibly even a tampered with clinical trial. The final nail in the cocoa flavanol's coffin is that there are these 2 pretty cool metrics that are called absolute risk reduction and number needed to treat. I like this idea that statistics has actually pretty neat system of checks and balances in place. Let's talk about RRR and ARR. Imagine we ran 3 separate clinical trials to measure MACE outcomes, and in all 3 of them we got the exact same distribution of events between 2 equal groups within a trial. Even though the relative risk was reduced equally in all 3 studies, in each trial, intervention group had just a third of MACE outcomes compared to the control. One group gained a crucial advantage to their survival from the intervention, whereas a different group saw very little benefit. To know which group benefited most from the intervention and to understand whether it has any rationale, any practicality, we have to look at such metrics as absolute risk reduction and number needed to treat. Think about it, almost 11,000 participants took cocoa extract every day for 3.5 years, $22 million spent. 76 people still died of CVD, 28 were spared. That's $800,000 to save 1 life. I'm not saying that 1 life isn't worth saving. All I'm saying that the effectiveness of cocoa flavanols is egregious. NNT is a cool metric that shows how many people need to undergo an intervention to prevent 1 adverse event. For CVD deaths, NNT of 385 means that 385 people will have to spend $44 every month for more than 3 years to save 1 life. You will have to pay $1,900 out of your own pocket. The COSMOS trial beautifully illustrates why NNT completely changes how we interpret clinical significance. When researchers rely on relative risk reduction to say 27% fewer deaths, it sounds like a breakthrough. The NNT calculation forces us to look at the denominator, revealing that for 99.74% of the people taking the supplement, it made absolutely no difference to their survival. Regarding major cardiovascular events, 3-point MACE. An NNT of 256 means that if you gather 256 older adults and have them take the cocoa extract supplement every single day for nearly 4 years, one person will be spared a major cardiovascular event. 255 people will derive absolutely zero objective cardiovascular benefit from the supplement. They either would have remained perfectly fine without it, or they had a major event anyway despite taking it. That is, if we take the results of the Cosmos trial at face value. This is such a fun metric, and it reveals absolute absurdity of even considering spending a single dime on cocoa supplements. It doesn't do anything to your health, but makes your wallet significantly lighter. There are also concerns that a 2x2 factorial design of the trial may complicate interpretation of the results, and actually introduces further statistical limitations, but that's too deep for me to discuss. All in all, if a scientist tells you there are certain cocoa flavonols save lives based on this data, they are violating the principles of their own discipline. You have read the data exactly for what it is, a highly unstable signal that proves nothing definitive without a massive high event replication trial. I wish somebody smarter than I am could back me up on this. This is what PhDs and MDs on YouTube should have explained to you, had they had your best interest in mind. But they were too busy sucking on fat corporate dick. Thanks for watching.